Well, hello everybody, and welcome to the America's Response to COVID-19 Pandemic webinar for legislative offices. My name is Stephen Rapundolo. I'm the President and CEO of the Michigan Biosciences Industry Association, or MishBio for short. We, along with our partners at the Healthcare Institute of New Jersey, at Biocom, at Bio New Jersey, California Life Sciences Association, and the Life Sciences Pennsylvania are pleased to present today's session, and I thank each of our partner organizations for their efforts in organizing the speakers and the topic. So obviously, under the circumstances, we can't be there on Capitol Hill as we have been over the last 8 years to host one of these briefings. The goal remains the same, though, with this virtual hookup, and that is to educate legislative offices on timely and relevant topics and leave you, quite frankly, with a better fundamental appreciation of some pretty complex subject matter as it relates to the biosciences industry. So the premise for today's briefing is pretty simple. Obviously, these are very trying times, no more so than the frontline first responders and healthcare providers that are dealing with the sick. But we wanted you to learn about the other front line. That is countless researchers across a variety of organizations nationally and around the globe are racing to discover and develop new treatments against COVID-19 and other emerging infectious diseases. We can just ask everybody to mute their lines. That would be marvelous.
An Easter surprise.
As a 20-year veteran of pharmaceutical research myself, and someone who helped develop probably some of the most recognized drugs on the market today, I can speak firsthand to the driven dedication of the scientists and other bioindustry professionals that are doing their best to discover and develop these new treatments. And for them, it truly is all about the patients and about changing lives. But good science and facts can't be forsaken for speed. Proving efficacy and above all, patient safety is imperative. And of course, that takes time. So you're going to hear from several experts from government, from large and small companies, from biopharma, from diagnostics, who are going to discuss how their organizations are working hard and in some cases collaborating to come up with the needed answers. So this will be just a small but hopefully insightful glimpse of the bio-industry's overall effort to combat COVID-19 and emerging infectious diseases. And lastly, before we get started, a couple of housekeeping notes. We're going to have some time after each speaker to field a few questions. You can submit those online through the chat question box, and we'll pose as many as time permits. In addition, the webinar is being recorded, and we'll have a link to share for those who cannot attend or if you wish to view the webinar again. So thanks for joining us, and we hope to see you soon in person on Capitol Hill and in our state capitals. And now it's a pleasure for me to introduce to you our first speaker, Dr. Rick Bright, who is the director of the Biomedical Advances Research and Development Authority, otherwise known as BARDA, and he also serves as the Deputy Assistant Secretary for Preparedness and Response at the U.S. Department of Health and Human Services. So Dr. Bright, welcome. We appreciate you spending some of your valuable time today. I'm sure you must be amazingly busy. I'm going to go ahead and switch the presentation over to you. Where are we? There we go. All right, it's all yours. Oh, hold on, Rick. Let me unmute you. Sorry. Rick, you're self-muted. There
Okay.
you go.
I've never been self-muted before, but and now's a good time to start probably. But I thank you very much for hosting this important forum and giving us an opportunity to give such important updates in the middle of such an important outbreak for our country and for the world. I appreciate the opportunity to give an update of what BARDA is briefly and most importantly, what we're doing right now with our industry partners to be able to position ourselves to respond quickly and effectively to this outbreak. I can't see the slides, so
I'm trying to figure out why not.
I can see a lot of handsome people.
Yes, indeed.
Nice. There we go.
But that is, oh my. Well, we seem to have lost you as a presenter for some reason. Let me,
This is the beauty of telework.
and that and so much for having a PhD. Doesn't, yeah, it's interesting. I've got a whole presenter list here and you're missing even though I've got you on video and I don't understand. Never fails. You go through the run-throughs and they're always perfect, and then the minute you go live. All I can do, Rick, is to turn it back over to me, and we can go through the slides, and I can advance them.
Perfect. We can do that.
Sorry about that.
We are flexible. So the next slide, I want to highlight for those of you who are not familiar with the Assistant Secretary for Preparedness Response, or the ASPR, that we are an agency within the Office of the Secretary in the U.S. Department of Health and Human Services, and we have one mission, and that mission is to save lives and protect Americans from the 21st century health security threats. On the next slide, it's an important statement that most of you are familiar with, that we are living in an increasingly complex and an increasingly dangerous world. We are seeing threats from state actors that might involve nuclear threats, or we're seeing threats from nature or terrorists as well that might include other explosives or anthrax or fentanyls, or as concerningly as we're in the middle of now, a threat from nature where we have a pandemic outbreak of the coronavirus. So BARDA is established to be able to respond to each and every one of these threats, to make sure that we are prepared with an array of medical countermeasures that include drugs, vaccines, and diagnostics to be able to combat each of these threats. Our next slide. Well, there's an example of many of those threats. So as the wheel of threats actually that we are concerned with, and we are grateful to be able to establish to address those threats. The BARDA models, BARDA is the Biomedical Advanced Research and Development Authority. We have a key role in government to bridge government with our private industry partners or private sector partners to accelerate the availability of medical countermeasures by forming unique public-private partnerships. BARDA doesn't just provide funding to companies to develop a vaccine. We also bring to the table a lot of resources, including expertise from the industry itself that includes hundreds of people within BARDA who have 10, 20, 30, 40 years of industry experience in every facet of making a drug or vaccine or diagnostic, building a facility, validating it, working through the regulatory processes, so we can actually join our companies hand in hand to accelerate the development of medical countermeasures. Our next slide gives you an example of many of the partners that we work with in industry. Today, we work with over 300 different companies to achieve our goals of developing life-saving drugs and vaccines and diagnostics. On the next slide, it goes to show that we also work with our government colleagues. We can't be one single entity and one single agency working with industry to accomplish the goals. So we also work hand in hand in coordinating across HHS, working closely with FDA in particular, also across almost every other department in the US government that has any role in developing or evaluating or using a medical countermeasure. It truly is an all of government approach, an all of industry approach, and a public-private partnership approach to accomplish our goals. The next slide gives you an example of some of those successes. So in our 13 years of existence in BARDA, we work with those industry partners and our government partners to achieve now over 54 FDA approvals, licensures, or clearances of drugs, vaccines, and diagnostics, and other enabling technologies or facilities to be able to make those medical countermeasures so they're available when we need them. The next slide, however, brings home a very important lesson and message that we deal with every day, but most acutely to what we're dealing with today. Speed is a critical component of any response. So even if you have the most advanced countermeasures, they will fail unless they are present in sufficient quantities with minimal delay at the location of need. So it's critical as we think about responding to something that's fast moving as the coronavirus, that we are putting all technologies in play, all companies in play, all knowledge in play to be able to respond as quickly as possible. In our next slide, we have talked about the strategy of approaching an end-to-end solution for a number of years. It is important that we innovate in every aspect of recognizing something new that has emerged, recognizing who is exposed or infected by that new pathogen, recognizing the need to design a medical countermeasure as quickly as possible, validate it through non-clinical and clinical studies, produce it at large scale, distribute it, administer it as quickly as possible. This is traditionally a 10 to 20 year timeline that we are asked to recognize the importance of every one of these steps in parallel during an emergency response and condense that 10 or 20 year timeline into as little as 1 year or even less to be able to respond to something as fast moving as the coronavirus outbreak. In our next slide, it leads us to the strategy that BARDA has been putting in place to respond to the coronavirus. First of all, we must accelerate everything that we're doing in parallel, leveraging existing partners, leveraging existing platform technologies with a track record of responding quickly to be able to protect Americans and save lives. We have to also recognize risk and mitigate that risk and monitor that risk as quickly as possible as it arises, as we can anticipate it, so we can be able to address that risk and move forward as quickly as possible. We have to prioritize investments that are based on the greatest public health impact and the probability of success. And we have to plan for success. We cannot wait for one clinical trial to finish before we get the data and plan for the next clinical trial. I cannot wait for clinical trials to finish before I start scaling up manufacturing. I cannot wait to plan a manufacturing run before I start ordering raw materials that might take 6 to 9 months to get in sufficient quantities. So we must plan for success from day one from this outbreak. We must build out redundant manufacturing capacity. We must mobilize our entire industrial base to work together collaboratively And we must think of every raw material that's required for mass campaign of therapeutics, diagnostics, and vaccines to have all of those supplies in place when the vaccines and drugs and diagnostics are ready. So it is definitely not business as usual. In our next slide, I give an example of some of the areas that we are working now with our industry and government colleagues to accelerate. We're looking at parallel work streams, not assuming one company is going to be the successor, but maybe multiple companies will be the successor. Multiple companies can play a role in helping to fulfill large volumes or large capacities for one or two successors of a vaccine or therapeutic. We have to work very closely with our regulatory authority and the regulatory frameworks to be as flexible and nimble as possible while ensuring full safety in every aspect and full recognition of any risk that we are assuming, and to be able to monitor very closely for those risks in real time. We have to use flexible contracting tools. So we have, in the last several years in BARDA, been exercising many more flexible contract tools, such as other transactional agreements that allow our partners in industry to work as a consortium to bring new technologies or new enabling supported companies into a portfolio rather quickly. We were able to modify these in a matter of days at the outset of this outbreak to be able to put those partners and those tools in place quickly instead of negotiating a traditional contract for the next 6 to 9 months. We also have to access core services such as clinical specimens, animal models, and all reagents and assays that will be required in real time to be able to support the development of drugs and diagnostics and vaccines as they're developed. On our next slide, we talk about risk mitigation. So I think we might have skipped a slide there. We talk about risk mitigation. So risk mitigation means I cannot pick a winner right now. None of us can actually determine which vaccine, for example, will be the most promising or will be the safest and most effective vaccine or will be available in enough capacity to respond to a global outbreak. So we must put several different technologies to work quickly and plan for success of each of them. We also must look at multiple diagnostic platforms and approaches to diagnostics. We started by working with the most experienced manufacturers in centralized laboratories and with experience in high throughput testing. And our strategy has been to move those diagnostics closer and closer and closer to the patient with shorter and shorter and shorter, faster turnaround time, so people and their healthcare providers will know who is infected or who has been exposed to this virus. We also need to look at the way we develop drugs and put drugs into clinical trials. We believe that we must lead by strong science with randomized controlled clinical studies that give us the best data to be able to make informed decisions on how we will respond with a therapeutic to this response. And again, I've already talked about the raw material supply chain. We need to start bringing those supply chains to the factories as soon as possible to be able to have those supplies ready when they're needed for mass scale. The next slide is our domestic manufacturing capacity. So again, it is one thing to have a wonderful drug or a wonderful vaccine, but if it's made by a single company with limited capacity, then we're going to find ourselves with a significant bottleneck, a significant public health challenge, and a significant communications challenge for the world to know that we have something that works, but yet we hadn't thought about manufacturing it at a large scale. So these are activities that must happen now, even before we begin some clinical trials of some of the drugs and vaccines that we believe might work. We must scale up and we must scale out. We must encourage industry to make partnerships that they haven't made in the past, teaming agreements, plan for redundancy, plan for producing a vaccine or a drug in multiple facilities at once at this point. We must look at the tech transfer opportunities and challenges and assay development that lie before us and start those tech transfers to those industry colleagues and partners today. We must build on and collect the raw material supply chain, make sure that is secure, make sure those are in place at the factories that will be doing this in a redundant manner today. And we must think all the way through, not just production, but how we will fill, how we will finish, how we will vial or deliver, administer that vaccine or that therapeutic today, so we have those supplies in place for a mass campaign as quickly as possible. The next slide talks about our interagency collaboration. So there are lots of ideas, lots of solutions, lots of potential solutions that we are learning about every day from entrepreneurs and companies, large and small, from academic institutions, from stakeholders around the world. We have established a single port of entry that will reach every one of these governmental agencies to be able to review those ideas, those solutions as quickly as possible. www.medicalcountermeasures.gov. It is not effective to submit your ideas to every single one of these agencies independently or drop it into someone's mailbox. Mailboxes are really overloaded because of all of the assistance that is coming our way. So we ask you, if you have ideas or hear of ideas, to funnel those through this medicalcountermeasures.gov website, and each of these agencies have real-time access to those ideas, and we'll review those, we'll prioritize those, and we'll follow up quickly. The next slide is about that industry engagement. So this slide was made on March 30th, and so actually it's outdated even from the date this was printed. We've had over 1,800 different ideas submitted to this portal today. We're reviewing those quickly. We had over 165 of those Tech Watch meetings or one-on-one conversation with those technologies. So the system is working. We are reviewing your ideas, and we are really impressed with the number of ideas that are coming from every level of entrepreneurship and capability across our country and across the world. BARDA has active partnering opportunities, solicitations available today. I'll show you the website on the last slide coming up. But in any area for medical countermeasures, vaccines, drugs, diagnostic therapeutics, we have a solicitation. So if you go through our portal process, you're reviewed, you're prioritized, we will direct you to those solicitations for rapid review. Next slide, please. This is the portfolio that we've built so far today, and this is changing by the hour every day. A number of therapeutics, a number of different approaches to either treating someone who's infected with coronavirus or treating the symptoms in late stage infection, or maybe even creating prophylactic approaches to prevent someone from getting infected. We're very pleased with the number of responses we're getting in therapeutics and also our vaccines and diagnostics areas, as well as different supporting efforts that we're seeing for animal studies or manufacturing or other enabling technologies that will allow us to scale these vaccines, drugs, and diagnostics as quickly as possible. Next slide, please. This is the hardworking group. This is not a very large group, but this is a group in BARDA that's making a lot of magic happen. This is our secret sauce. There's about 250 people in BARDA that are working day and night with our industry colleagues and our government colleagues. And my hat goes off to them, my heart goes out to them, and all the relationships that they're forming and building to make sure that we are reviewing and accelerating the development of life-saving medicines every day. The next slide, please. This is my challenge to you. So those of you who are familiar with a challenge coin, I want to present this to you out there across Congress and all of your stakeholders. Normally, these are given out at the end of a successful milestone as an acknowledgement of the challenge that's been met. At this point, I'm handing this out to each of you to take this challenge, find solutions, find new ways of working together, plan for success, do everything possible to save lives in this outbreak. Next slide, please. Final slide. This is how to reach us, how to contact BARDA. Go through the portal, submit your ideas, funnel any ideas to that portal, look at our solicitations, and we're not too afraid to ask for help. So if you know people out there who have expertise in this area, who want to join our family, want to join the fight, there's a listing there as well to submit their resumes so we can hire assistance as quickly as possible. It will take every one of us in Congress, in government, in industry, in society to beat this, and we will do it. Thank you all.
Thank you, Dr. Bright. Excellent, excellent. And thank you for you and your group's immeasurable efforts. I can't even begin to imagine what you folks along your days are and what you must be going through. So thank you. Let's see if there are, oops. Let's see if there are any questions. I don't see any. So why don't we move on to the next presenter, and that's Dr. Kate Broderick. She's the Senior Vice President of R&D at Inovio Pharmaceuticals, and also is heading up the project, Inovio's nCoV project. Inovio is a small Pennsylvania-based biotech company that's focused on the discovery and development of synthetic DNA products for treating infectious diseases and cancers. Dr. Broderick, thank you. I'm going to switch you over, hopefully this time without any issues. I think you have control. On Dr. Broderick, just one moment here. There we go. All right, you're unmuted and we see your screen. It's all yours.
Excellent. Fantastic. Thank you so much. And thank you so much for the opportunity to speak here today. It's a real honor. And to be followed by Dr. Bright. And Dr. Bright, if I could convey it, was really nice to hear an optimistic note to the end of your presentation. I think all of us involved in this situation at the moment, I think we can all be kind of overwhelmed with the task before us. So it really is good, I think, for us all to kind of coalesce around. We will find solutions for this, and we will find a path forward here. And if I might just very briefly note, the solidarity that I've seen in the scientific community over the last few months has been absolutely remarkable. And it's really, if there's a single positive thing to come out of the situation we're in today, I think it is our ability to all work together towards a single goal, and I think that's really been fantastic. So as Stephen noted, I'm representing the company I work for, Inovio Pharmaceuticals. Yes, we're headquartered in Philadelphia, but I have the bad luck to be based in California, in San Diego. So we really from day dot, as I'm sure you all were very interested in this outbreak. I remember very clearly on the 31st of December reading about it on the news, this odd outbreak of pneumonia, and really immediately tracking it. And then for us, the trigger point was the 10th of January when we got access to the genetic sequence of the virus. So we have been leading DNA medicine technology for many, many years now, and have worked extensively on Zika, on MERS, on Ebola. And so really, I think our technology platform has really put us in a position where I think we're positioned very nicely to respond to the outbreak that we're all experiencing today. So let me kind of go over some of the high level background here. I don't have a whole lot of slides, so I might talk a little bit more in detail than what the slides outline. So really, I think, we could all see the urgent medical need, specifically from our perspective, for a vaccine for COVID. And so we immediately decided that we wanted to apply our DNA-based vaccine technology to address this critical need. And I'll go through where we are today. And I really, even in my own head, when you think, we only got access to the viral sequence on the 10th of January, we're going to be in the clinic in April. And it's hard for me, my own self, to really wrap my head around the rapidity of that response and, many of the other responses of the other technology platforms. It's really quite remarkable. So one of the main areas I think that make our DNA medicine technology so amenable to the situation we're in today is our accelerated candidate discovery. And just an example of that, we received the genetic sequence from Chinese authorities, and 3 hours later we had designed a vaccine against COVID-19. So really we were able to respond extremely quickly. We were able to test small-scale manufacture and test multiple candidate constructs very, very rapidly. In fact, we were in vitro and in vivo testing within 14 days. We've subsequently transferred our candidate now over to our GMP manufacturing lot, and obviously that's now completed and will be used in our clinical trial, which is starting in April. And we've also had the good fortune, I guess, or perhaps you might say the good planning, to be able to leverage our regulatory precedents to support our entry into the clinic. So the FDA, the EMA, and many other authorities are very familiar with our platform. And so, for instance, didn't require a toxicology study in advance. We're very familiar with our safety profile in the clinic and our manufacturing technology. So almost we were able to kind of provide them with all that documentation up front that they had already reviewed many, many times for our other programs. Another thing that I think was extremely beneficial from our perspective was our previous work on MERS. We actually have run a phase 2 trial for our MERS DNA-based vaccine candidate, and that was in partnership with Samsung in Korea. We've also partnered with CEPI for our ongoing phase 2 trial in the Middle East and Africa. So we were able to use a lot of our learning from our MERS program to apply to SARS-CoV-2. Now, obviously, as we all know, the homology wasn't enough that we would be able to use the same vaccine, but I think the knowledge garnered from our challenge studies for MERS, where our primates are 100% protected from challenge with MERS, and the clinical readouts that we really help us design our clinical trials, design our challenge studies, so that we were in a preferred position moving forward with our vaccine development against COVID-19. So this is just a snapshot of the preclinical work that we've done so far. For this, we've actually now tested our COVID-19 vaccine, which is called INO 4800, in mice, guinea pigs, rabbits, and primates. The rabbits and primates, of course, have the benefit that you can give them the full human dose. Guinea pigs are a particularly nice model because their skin reflects very well the skin physiology of humans, and we're actually going to be using an intradermal administration for this vaccine, so we'll be delivering it to the skin rather than to the muscle. And really just at a high level, we're very happy with the immunological responses that we've seen in all of the animals that we've tested so far. And what's particularly encouraging from my perspective is that we're seeing those responses very, very rapidly. So you can see here the responses after day 7, after day 10, single immunization. So we're getting really robust T and B cell responses in all the animals that we've tested thus far very, very rapidly. So we're very excited about that. And that reflects very well what we've seen so far for our MERS vaccine too. So we feel that what we learned there, we can definitely apply here. Some of this data has been submitted to Nature Communications and is undergoing review at the moment, and we'll be publishing the rest as soon as it's available. We're also partnering globally to perform challenge studies against COVID-19. We'll be assessing that in both ferrets and in primates. And one of our collaborators is Public Health England at Porton Down. And so we're very excited about getting efficacy data, which of course will be vital as we want to push forward to phase 2 testing. But we'll also be answering the questions regarding vaccine-enhanced disease. And I would like to point out that we saw no evidence of that whatsoever in any of our MERS challenge data for the primates, or of course in the clinical studies either. So we feel confident that that will be the case here. But I think it's an important box to check, and one that obviously we have to absolutely focus on from a safety perspective. So those challenge studies are underway, and will be really pivotal data, I think, to add to the package as we move forward. So just a little bit about the product presentation for INO 4800. That's our DNA-based vaccine against COVID-19. It is going to be a 100 microliter injection, and that's going to be, as I mentioned, injected intradermally. Then we use our smart enhancing delivery technology, and we're positioning for this particular portfolio our 3PSP device. So just I won't spend a ton of time on this, but just to give you a visual, it's about the same size as an electric toothbrush. You just take the device, you apply it to the area that you've injected your vaccine, and really it just absolutely improves the uptake of the DNA into those skin cells. So it's a very brief process, a very brief procedure that's very well tolerated in the clinic. We're going to be supplying our vaccine in 5 ml vials, specifically for use for stockpile scenarios. And we're also looking at multi-use vials for consideration, obviously, for pandemic situations. One extremely positive characteristics of our product is the stability. Our storage profile long-term is at 2 to 8, so refrigerated, but our product is stable at room temperature for a year and stable for approximately a month at 37. So really, from a storage perspective, I think we're meeting all the criteria there. And our expected shelf life is greater than 4 years. So just a little bit about the regulatory paths that we've been moving through. Our first human clinical trial, as I mentioned, will be starting in April, and that's going to be a 2-site US-based trial at the University of Pennsylvania and also a site in Kansas City. So of course we have been and are in discussions with the FDA in regards to that. And we've also been speaking to the EMA and also the Korean authorities and the Chinese authorities, because in addition to our US-based trial, we're also hoping to run parallel trials in Korea and China, just following the initiation of our phase 1 in the US. As I mentioned earlier in the run-up, we were able to leverage much of the previous regulatory knowledge that we've amassed over many years now with the FDA in regards to our plasmid manufacturing, our non-clinical and clinical safety database, and also our device master file. So when it comes to clinical testing, we were very grateful to be awarded $9 million from CEPI to start our INO 4800 program, and that's going to cover our phase 1 trial. The Gates Foundation also has awarded us support for this trial as well. We're very, very grateful for their support. We're starting that first clinical trial in April, but we hope to really kind of scale up to phase 2 as soon as possible following that data. And obviously we will need additional funding support for that. We're also looking at starting trials, as I mentioned, in China and Korea, and we're already in discussions about how to expand what we're doing right now for appropriate efficacy studies. One of the strong elements of DNA medicine is the scalability of manufacturing, and we were able to announce a collaboration with Ology. So that's a US-based manufacturer to manufacture some of our product. We're also working with large-scale manufacturers across the globe, and have set ourselves the internal goal to have 1 million doses of our vaccine ready this year, but are already talking about the scale-up required to take that to population levels. And that's obviously going to require multiple manufacturers working in parallel. And that's really underway. Those discussions are underway at the moment. And that's my last slide. So I am very happy to take any questions or to clarify any points moving forward. Thank you very much for listening.
Thank you, Dr. Broderick. Let's see if there are any questions. I do not see any. So we're going to move on to the next speaker, which is Dr. Rick Nettles. He's the Vice President for U.S. Medical Affairs in Infectious Diseases at Janssen Pharmaceutics, a division of Johnson & Johnson. Some of you may have heard the news yesterday. J&J announced a lead vaccine candidate for COVID-19, along with a new landmark partnership with BARDA to combat the disease. Now, Dr. Nettles is on a phone line and does not have slides, so we're simply going to put him on audio as soon as I unmute him. Okay, Dr. Nettles, you should be on.
All right, Stephen, can you hear me okay?
Yeah, yes, I can. Hopefully the attendees can too.
All right, well, fantastic. Good afternoon. Thank you for inviting me today. It really is a privilege to be included in this panel of speakers. So I lead medical affairs, as you mentioned, at Janssen Infectious Diseases, and that is part of the pharmaceutical companies of Johnson & Johnson. So I'm really here to represent J&J, and I will speak to the vaccine news that you just mentioned, but I won't go in as granular as detail as Dr. Broderick did. Rather, I'll speak broadly on the response of J&J as a company as a whole. Before I get going on that part of the discussion, I just want to take a minute and send our thoughts out to the thousands of people around the country who have been infected and families who are dealing with loved ones. As a global company, our J&J employees are based in Italy and Spain, and I can tell you we're really feeling the impact now. And finally, as an infectious disease physician, my thoughts go out to the frontline providers, my colleagues and friends who are in danger right now. With that said, J&J has a 134-year history, and we really feel like our company can quickly mobilize to bring resources to this epidemic. At the outset of the disease, we took a multi-pronged approach with regard to our R&D efforts, and that's what I'll talk about first. We have mobilized our efforts along the lines of developing a vaccine, and then also screening a library of antiviral compounds. And you saw that on the slide that Dr. Bright showed from the BARDA presentation. Yesterday, we were pleased to announce that we selected the lead COVID-19 vaccine candidate from the constructs that we've been working on since January. We are using the same AdVac platform that we have to develop an RSV and HIV vaccine that's in phase 3 right now, a Zika vaccine, and then an Ebola vaccine as well that is in late-stage clinical trials in Africa today. So using that platform and the construct that we've selected, we are moving forward with our lead candidate. With the announcement yesterday, we also announced that we've significantly expanded the partnership between Janssen and BARDA to rapidly accelerate the research and development and human testing of our vaccine candidate. This is really a landmark partnership for us that commits to more than $1 billion of additional investment between BARDA and J&J, and we are very, very grateful for that collaboration with BARDA. As part of the commitment, J&J is also expanding the company's global manufacturing capacity, including, importantly, establishing new U.S. vaccine manufacturing capabilities, and we will also be scaling up capacity in other countries. So with this additional capacity, this will assist us in rapid production of the vaccine and will enable the supply of more than 1 billion doses of vaccines globally. With regard to the timelines, the company expects to initiate phase 1 human clinical trials by September of 2020 and anticipates that the first batches of the vaccine would be available for emergency use in early 2021. The company plans to imminently begin production in order to have that amount of vaccine available even before the phase 1 clinical trial begins. And we expect to bring forward an affordable vaccine to the public on a not-for-profit basis for emergency pandemic use next year. Johnson & Johnson and BARDA will co-fund the vaccine research and development, including the preclinical, the clinical development, and upscaling of the clinical activities. In addition to the vaccine work, we're expanding our partnership with BARDA on upscaling Janssen's ongoing work to screen compound libraries to include compounds from other pharmaceutical companies in addition to J&J, with the aim of identifying potential treatments against the novel coronavirus and progressing them up through phase 2B. So in this approach, we're also screening not just compounds that are in development, but also compounds that are already approved for other uses. The thought behind this approach is that if we can find a compound that is already approved for other uses, it's known to be safe, and we show that there's a signal for potential efficacy against the coronavirus, we would immediately begin to use that in the clinic to treat rather than having to progress it through clinical trials. Our partnership with BARDA predates the coronavirus pandemic, with J&J Innovation announcing a collaboration with the organization in 2019 to develop and maintain specialized innovation zone at our newly announced J-Labs facility in Washington, D.C. So Johnson & Johnson J-Labs is a global network of open innovation ecosystems in which we collaborate, bringing forward solutions in the pharmaceutical, medical device, consumer, and health tech sectors to create and accelerate life-saving solutions. The Washington, D.C. J-Lab Innovation Zone will be dedicated to the advancement of medical countermeasures aimed at securing our nation from chemical, biologic, radiologic, and nuclear threats, as well as from pandemics such as influenza and other emerging infectious diseases. Beyond R&D, I'd like to stress that J&J has taken other measures during this pandemic. So, for instance, we have a robust business continuity plan in place across our global supply chain to prepare for events like this. This includes maintaining critical inventory at major distribution centers away from high-risk areas and working with external suppliers to support our preparedness plans. As part of this, we're closely monitoring our product demand and supply levels across the U.S. and global network to ensure adequate and effective distribution and working diligently to make sure that we're meeting patient, consumer, and customer needs. We're also taking precautions to support the safety and well-being of our employees and contractors and the communities in which we live and work, and mobilizing to provide equipment, products, and financial donations to support organizations and healthcare workers. So as an example, in January, J&J announced support of frontline healthcare workers through our J&J Center for Health Worker Innovation, and we committed $250 million over 10 years to this fund. Last week, J&J increased that commitment by $50 million for immediate COVID-19 response, primarily focusing on frontline healthcare workers. Cash donations from this have been made to various organizations, including the COVID-19 Solidarity Response Fund, CDC Foundation's All of Us campaign, American Nurses Foundation, the Swiss Red Cross, the Red Cross Foundation, Wuhan branch, and the Community Foundation of New Jersey Disaster Relief Fund to support the patients who are most vulnerable at this time. We've also introduced a variety of employee policies to help address the pandemic. So, for example, we now have a medical personal leave policy, which enables employees who are medically trained personnel to access a global paid leave of up to 14 weeks through March of 2021. And we've worked to convert some manufacturing lines at several facilities around the world. Some examples of this, we are now converting lines to produce hand sanitizer so that we can protect our R&D, manufacturing, and distribution employees, and also look to be able to donate bottles of the internally produced sanitizer to communities on the front line. Another example was given by Alex Gorski, the CEO of J&J yesterday on NBC when he was asked about the supply of Tylenol. So an example of streamlining the production and eliminating some of the formulations of Tylenol so that we can rapidly produce as much of the basic formulation of Tylenol as we possibly can. So J&J aspires to be a leader in the global health commitment to combating infectious diseases. We have been working against TB, HIV, Zika, and Ebola over the last couple of years, and we see this coronavirus as another example where we have a great desire to step up and make a difference. I would just like to end by saying we want to thank the partners that we have been engaging with and are open to all collaboration from partners or pharmaceutical companies around the world to deal with this coronavirus pandemic. Thank you.
Thank you, Dr. Nettles. Any questions for Dr. Nettles? Before we move on to our last speaker, there is a question that I neglected to pose earlier. It's directed to Dr. Bright. Let me just make sure that he is unmuted. All right. The question reads, Dr. Bright, can we afford to move at the speed of government when we have hundreds, if not thousands, of people dying from the virus every day? In Michigan, we are forming a registry to keep track of various treatments for very sick patients. This will be a coalition of government insurance hospitals and universities into our registry to evaluate various treatments. Would you have suggestions for off-label or compassionate use in this population?
I actually am interested in the comment, can we afford to move at the speed of government? I've seen the government move in unprecedented speed and unprecedented collaboration in this outbreak, something I have never seen this government or any government do for any prior outbreak. And that is only because of the ability to learn from lessons of the past, from the Ebola outbreaks, from the Zika outbreaks and MERS outbreaks and other pandemic influenza outbreaks. So I hope that it's obvious to everyone listening and obvious to the world that this is not business as usual. The speed of government right now is quite impressive when it comes to medical countermeasure development and acceleration and partnering and reviewing every possible solution to this outbreak as quickly as possible. I do understand that there are a number of compounds that individuals or groups or companies believe might be useful in this outbreak or useful to treat coronavirus. And I urge a lot of caution to proceed cautiously if there's no randomized clinical trial to support claims or anecdotal evidence or observational evidence. All of those types of evidences are important and critical. And our FDA is working very quickly to review those as they come up. Our medical countermeasures working group task force, we have a special working group on clinical trials, special working group on therapeutics. They're reviewing those as quickly as possible as well. So if there are suggestions or potential treatments that are out there that are viable, I encourage anyone to direct those into the portal. I encourage them to make sure the FDA is aware of those. And our colleagues at NIH and CDC and BARDA are doing a lot to move quickly to evaluate those, candidates and put those in an RCT as quickly as possible. I do not encourage off-label use of medicine that is not carefully monitored and reviewed by the FDA and healthcare providers at all. I urge caution in each of those areas. We can get the data, we can get data quickly, and we do have clinicians and FDA and regulators reviewing those as quickly as possible. And that is the path I prefer that the world follows as we try to find a solution and treatment as quickly as possible.
Great, thank you very much, Dr. Bright. I don't see any further questions, so we'll move on to our last speaker, Joshua Trotta. He's Senior Director of Genetic Sciences at Thermo Fisher Scientific. So while the previous two speakers focused on the therapeutic side of the topic, Joshua is going to speak to the diagnostic testing for COVID-19 side. Joshua, I'm gonna switch the controls over to you and make sure that I unmute you. There you go. You should be ready to go.
Okay, very good. Well, thank you all. It's a pleasure to be able to join the conversation this afternoon. As was stated, my role today is to represent just a diagnostics viewpoint of how, of all the news that we're hearing on a regular basis about testing and the needs for testing, and specifically just representing Thermo Fisher Scientific and the role that we play. So I'm going to step back a little bit just to talk a little bit about how the diagnostics are actually developed and some of the paths that are taken. Also address just kind of common questions that I think are often brought up about, you know, what is the test? What is actually incorporated as part of this? What are the workflows? Like, what are the steps that are actually needed? I know people talk about it broadly, but I'll speak a little bit just about those steps and those workflows, and then also speak just about the scalability of some of these solutions and some of the next components. So again, as stated, I represent a business development team specific to the COVID response. Our group was the one that had designed both solutions that are multiplex assays, and we'll talk about what that means, as well as for Thermo Fisher Scientific is a global supplier. And I think as we've heard from some of the previous speakers, you know, we have participated in areas like BARDA programs supporting, you know, radio biodefense programs, anthrax, being able to be a manufacturer and a supplier of both instrumentation as well as consumables and the entire workflows to kind of support the global needs in diagnostics, specifically the area that I'm focused on, but we also have colleagues within the organization focused on the vaccines, personal protective equipment, and kind of broader ranges of kind of the needs across the globe. So with that, I'll step back just on more of a basic discussion point about coronaviruses. You know, as we think, I think the audience, it depends on who's there. So for those that are epidemiologists, or this is where you live and breathe, excuse me for just the simplicity of this one. But when you look at, there's different types of coronaviruses that are out there in the market, as we've heard about from both MERS and SARS of previous viruses. This coronavirus that we're dealing with now is a novel coronavirus. So as you look at the spread and how these viruses have happened, we heard, I think earlier from Dr. Broderick, of just the information that becomes available once somebody is identified with this virus. And that generally starts with looking at next-gen sequencing. So looking at the genome of the virus, of identifying, is there something when we genetically analyze this information, is there any pathogen that's known in the world to exist that matches this? And in this particular case of being able to identify that, it narrows things down into coronavirus. but there's something novel, there's something unique about this particular virus compared to previous ones. And so what that allows groups like Thermo Fisher to do is that we will then take that knowledge, it's kind of going from a broader Google worldview and kind of narrowing yourself down, if you will, into a street view and saying, instead of broadly looking at something over and over from a global perspective that costs a lot of money or maybe takes a lot of time to run that testing, if we now know that we want to look at a specific street and do that routinely, how do we do that efficiently? So that moves us into leveraging tools that are called bioinformatics to develop then assays or primer and probes that look at that specific genome of the virus so that we can identify it over and over again. And so not only to do that over and over again, but to do it with robust tests that are well known. In this case, qPCR technologies that's been out on the markets for over 25 years, and to develop cost-effective and robust tools that can then be scaled and deployed. So in this particular case, the approach that Thermo Fisher actually takes is there's a lot of ways that you can design tests and targets. Knowing that it's a virus and viruses have a tendency to mutate, we took the approach of actually designing targets across the genomic information, across areas that are known to avoid a mutation region. So if the virus were to mutate, we took a strategy of just looking to design our assays to still make the test robust, even if there there's a shift in the mutation itself for the coronavirus. So with that, we've kind of really reduced we're taking some steps to reduce the potential causes and making sure that the assay is consistent and robust. So with that, we move on then to say, well, what is that workflow? And so you probably heard and read quite a bit, but I thought I would just reiterate is really you start with a sample collection tool. And that includes a nasal pharyngeal swab. And this is for those that have never had a strep test, for the unpleasant experience associated with that. It looks like, in essence, a Q-tip that is, a very long Q-tip, although it's not a Q-tip. The top of the head is very different, but it's inserted into the nose. And what will actually happen as far as the sample collection piece is to then extract, in this case, the viral RNA from that, to mix it with some reagents that then move on to, which is the sample prep component, and then to be able to analyze that on a real-time or qPCR-based instrumentation, which you see demonstrated here. And what it's ultimately looking for is to say, across that sample that was selected, are we identifying the viral RNA? Is it present in the sample that was taken to determine whether somebody is positive or not? There's interpretive software that's also included in this workflow that helps, especially as we look at the scales that people are really ramping to be able to help minimize some of the just the manual processes using tools of software to help on the interpretation side. So an important piece, maybe just a highlight, I think for Thermo Fisher and why and kind of our role in this piece is that across this entire workflow, Thermo Fisher actually makes the tools. So from the sample collection piece through the sample extraction, the instrumentation that you see for real time, as well as the software, we make the entire solution and portfolio. I think what we've been able to do specifically in this particular response, as well as others prior to this, is that not only to be able to supply components to some of the large diagnostics testing facilities, as well as other global ministries of health, but to also be able to scale the volumes of testing. So currently, Thermo Fisher is actually making 10 million diagnostics tests a week that are utilizing the assay design that I've been talking about. With that, and I think it was highlighted earlier in this discussion by Dr. Bright, is the importance of having availability. So you can have the best solutions out there, but without having the availability of tools across each of this entire workflow, it doesn't do you any good. So our team internally, from a manufacturing perspective, R&D teams are fully dedicated to continuing to ramp the diagnostic solutions and the needs. We continue to look for ways to help expand as we look at the front end. Sample collection has been a recent pinpoint or a pinch point of looking at how do we address the scalability of nasopharyngeal swabs and viral transport medias. So the teams are working with a host of individuals across the FDA, the Bill and Melinda Gates Foundation, and others to actually do some clinical studies on promising potential tools that could help broaden the availability of these much needed nasal swabs as well as viral transport media. So we continue to focus and to drive holistic solutions to support the needs to help with the growing need of testing. So what does that mean? How does it look from a timing perspective? So there's quite a few, obviously, tests that are continuing to come into the market. So obviously I'm speaking to our EUA-approved version of the test, and what it uses is the 96-well plate. But because all of the markers to test for coronavirus are included in what we call a multiplex, so it's a single tube, it allows you to test 94 samples or patients in a run. And that allows you to do that every 3 hours on a platform. There's different configurations that allow people to scale more or to ramp your ability to do the sample extraction piece. But to give you an idea of just the throughput associated with this. So you can see here on the slide that the different types of swab types that are utilized currently, and then kind of the time frames associated with just the sample prep pieces, the qPCR piece behind this, and then the interpretation of this. To just give you a sense of how that actual workflow happens. So lastly, as we look about this, is just to design content that is robust, allows people to really scale the volume of testing, which we know is a continuing growing need. And there's other suppliers that are in the marketplace, but I think at 10 million, I would probably consider Thermo Fisher as a major supplier into the markets today to help, and we were committed to continue to do so. But also to make sure that we're continuing looking at the assay designs to make sure that it has a concordance to the genomes as more information becomes available, that if there are any mutations that our assays are 100% concurrent to the existing genomes that are on the market space. So with that, being able to then help our partners across the United States to really be able to, and across the globe, to really be able to respond effectively. So that's just a component, I think, of this broader presentation from today. And I think that's my last slide, so I'll take any questions that might exist on the diagnostics front. Thank you very much.
Thank you very much, Joshua. Appreciate that look on the diagnostic side of the world. Are there any questions for Joshua and COVID-19 testing? All right, well, seeing none, we're going to wrap up this webinar by thanking everybody who joined us, particularly the speakers. We appreciate your time. I hope everyone found the presentations to be informative. As a reminder, this has been recorded, so we will be happy to share a link out for it. Also, I just want to finish by saying this to the legislative staff that have been on the call today. Please do use your state bioscience associations as a resource. We're here to help you. We're here to connect you. We're here to educate you on an industry that is in each of our respective backyards, and quite frankly seeks nothing more than to improve quality of life for everyone. So in conclusion, stay safe, stay well, and thank you very much.
Thank you.
Thank you.